This video is an introduction to DNA, also known as the blueprint of life. And when we talk about a blueprint, typically we think about building a house or building some sort of structure in some way where you see the layout of everything that you're going to build. So in this case, what we can see is we have a very large house where we have a living room and a dining room and a kitchen and a hallway and a library and all kinds of stuff. Well, that's the blueprint on how to build a house. Well, DNA is a blueprint on how to build an organism. What does it stand for? Well, DNA stands for deoxyribonucleic acid, DNA. And it looks like this, where you have a spiral-shaped structure, and the structure can be coiled up into chromosomes, which then fit inside a nucleus of a cell if it's a eukaryotic organism. Some facts about DNA. DNA was first discovered, or the structure was first suggested by two people named James Watson and Francis Crick. So Watson and Crick provided the world the first model of what DNA looked like, and there it is right there. Here's James Watson, and here's Francis Crick, and here's the first model that they built. The shape is typically called a double helix, and it's also known as a spiral ladder. This is Rosalind Franklin. Now, Rosalind Franklin took an X-ray picture of DNA, and without her knowing it, it was provided to Watson and Crick. And they used that picture to help them figure out what the structure of DNA looked like. This was one of those stories in science history that is pretty important because she never got the credit that she deserved for first taking the picture of DNA in order to help them out. Now, what is DNA? Well, it codes for your genes or your traits. So back when we were talking about traits and genetics, DNA is the code to create those traits. And DNA is made of repeating subunits called nucleotides. And so here's a picture of DNA right there, spiral ladder. We have one side, it's actually a double helix, so there's two spirals going around each other. So here would be one side, and here would be another side that's being held together by hydrogen bonds in the middle. And a nucleotide is basically one of these units where you have a phosphate sugar and a base. We'll see that in the next slide. So what is a nucleotide? Well, it has three parts. There's a phosphate group. There's a sugar. The sugar is known as deoxyribose. And then there's what's called a nitrogenous base. Now, this is the really important part about DNA because it is the letters that make up the code for DNA. So one of those bases could be an A, a T, a G, and a C. So adenine, thymine, guanine, and cytosine are the proper names, but many people just call them by their first letter, A, T, G, and C. And so in a nucleotide, we have a phosphate sugar and a base, one of those bases, and that's going to repeat throughout. The bases won't necessarily repeat, but the sugar, phosphate, and some base will repeat. Now, base pairs, or there's a base pair rule, and if there's an A, adenine, it always bonds to a T, thymine, and guanine, a G, always bonds to cytosine. So we can see here this green base is going to be adenine, and that is going to bond through a hydrogen bond to thymine right here. And this dark blue is the deoxyribose sugar, and the light blue is the phosphate. So here's our phosphate sugar base. Down here we have a phosphate sugar base. So those are our repeating nucleotides. The sides of the DNA ladder are phosphate and sugar, and these are going to be held together by hydrogen bonds that go straight down the middle and connect the A's to the T's and the G's to the C's. So when we look at the base pairing rule, if we had one side of the DNA molecule that said ATA, TCA, TGC, and GGG, then the other side of the molecule, the other spiral that would be stuck to it through hydrogen bonds, would be the complement. And that would mean it would be TAT, AGT, ACG, and CCC, because A always bonds with T and G always bonds with C. So this is how they would always pair up. Now, a little more complex conversation would be that the A's and the G's are what are called purines. And we can see they have two rings. There's like a 6-sided ring here and a 5-sided ring here. That's the A. Same with the G. And then what are called the pyrimidines are T and C. They're only single ring structures. But the important part here is that a purine always pairs with a pyrimidine. And it turns out that the A, adenine purine, will always bond with the T, thymine pyrimidine, and the G, guanine purine, will always bond to the C, cytosine pyrimidine. Now, how does this code work? Well, the combination of A, T, G, and C determines what traits you might have. So, for example, this cat has purple hair. And in its DNA, it might say something like CAT, CAT, like cytosine, adenine, thymine, cytosine, adenine, thymine. That would give this cat purple hair. However, if it said TAC, TAC, maybe it would give it yellow hair. So depending on how the letters are sequenced, depending on the order of those bases, you're going to get different traits. And so that relates back to the genetics unit that we were talking about previously. So we can think of bases of DNA like letters of words. They are letters, but we can think of them like letters in words because letters form words, words form sentences, sentences form paragraphs, paragraphs form chapters, chapters can form books. And if we think of it that way, then every living thing is its own book. So the letters in the English language use 26 letters. The letters in the DNA language use 4 letters. However, they are put together in such a way that the different combinations of letters can create different combinations of organisms. If we want to get a little more detailed and kind of see how this might connect to chromosomes that we talked about during cell division, we can see that DNA coils in a very organized manner. And we first have our DNA, which gets wrapped around these proteins, and we call that chromatin. So this area right here would be chromatin. And then it coils some more. You can kind of see how all this DNA and protein starts wrapping around each other. And then this set of coils starts coiling around. And then those coils start coiling around until you finally come up with a chromosome. So if you ever wondered how DNA condenses into, or I should say chromatin condenses into a chromosome, well, this would be a good picture on how to visualize that. So as a review, what does DNA stand for? Deoxyribonucleic acid. What is the shape of DNA? A double helix. Who established the structure of DNA? Watson and Crick. And always remember, Rosalind Franklin helped them out. What does adenine pair with? Well, adenine, the A, pairs with thymine. And guanine would pair with cytosine. The sides of the DNA ladder are deoxyribose, the sugar, and a phosphate. Guanine always pairs with cytosine, as we said previously. And what would be the complementary sequence? CAT-TAG. Well, the complement then would be GTA-ATC. The two sides of DNA are held together by hydrogen bonds. DNA is composed of repeating subunits called nucleotides. And the four bases that make up the rungs of the DNA ladder are A, adenine, T, thymine, G, guanine, and C, cytosine. And so hopefully this short introduction to DNA has helped you understand basic structure and function of DNA.